All right, so I think we are good to start. So welcome again to our today's webcast, Master the Leap from Lab to Clinic. And that's it from my side. I wish you an interesting session, and I am handing over to our speakers who will introduce themselves.
Good morning, good afternoon, good evening, folks, wherever you are. My name's Gareth Duffy. I've been with Feder Pharma for almost 18 years now, and currently lead up our supply chain and project management team at our dedicated clinical filling site in Reinkweiler in Austria.
Hi, everyone. It's great to be here today with all of you. My name is Mark Rockhorst. I'm leading the project management and supply chain teams here in our Chicago facility in Skokie, Illinois. I also have been with Vetter for a while now, so for 12 and a half years. And I look forward to discussing a pretty interesting topic today. So Gareth and I are going to talk about what I think is one of the more critical steps in the development of a new drug product, and that is the shift from preclinical to for human use clinical manufacturing. Leading up to this, prior to the transition, a lot of attention and focus has probably been on things like small scale lab work, process optimization, maybe some formulation, biological activity, potency, safety, really science heavy focused work. But as you begin this transition to human clinical trials, this attention focuses changes more towards the processing. So you start looking at things like scale up, logistics, supply chain, regulatory impact, and a heavier reliance on CDMO partners. And I think this is a really important piece because if you get that correct early on, choosing the right CDMO to work with helps with a lot of those other things that your attention is now focused on, like your logistics, your regulatory, and the right partners can help support with that. So today, Gareth and I are going to dig a little deeper into some of these topics and highlight some of the things that you can expect through this process. We're going to talk a little bit about priorities and planning, as well as some important decisions you might be faced with along the way, questions you should be asking potential CDMO partners, and ultimately how to better plan and prepare for the long-term success of your new drug product candidate.
Okay, but yeah, let's start with planning. So what are the key steps that really do enable you to be successful when planning that first clinical trial material fill? You really should treat each step along this way as a stage gate and kind of re-evaluate your success along the way. So once you've selected your partner for your drug product, you've gone through that drug substance selection piece, you're now in a position to kick off the interaction with them to really start the activities that will eventually lead to you having a released batch of material. So establishing that relationship up front is key. And then they're going to want to know a lot of information from you. And these decisions can be key to guide your success moving forward. So we're talking about things like the material requirements and specifications. What do you know about your material? What do they need to know about it? You need to select that primary packaging material. Once you've done that, you need to define really your design space around that, develop a process with your partner together to get that product into that primary packaging container system you've selected. Then find a way to test that to make sure you're getting your quality attributes met to release material that is effective in the clinic. Go through some risk mitigation steps along the way with development or technical batches to really, kick the tires of the process. Make sure you really do de-risk that GMP batch that is so key along the way. And once you filled it, the journey doesn't stop there. It's been a long way to get to that GMP batch fill. But after that, there's a lot of activities that are triggered by that. So after that, we get things like the regulatory documentation support, getting that IND drafted, making sure that's concise and clear for whatever agency you want to hand it into. Once your testing is being performed in parallel, you'll eventually have released material, hopefully an approval from a clinical trial sponsor that allows you to get that product into the clinic and start the next stage of your adventure. So the first step along there is really just making sure that your API, the drug substance you've spent a long time developing is really ready for the next step, ready for that graduation step to move from the safety of a lab environment into what can be a very scary manufacturing environment. And a couple of questions you need to ask yourself along the way. You want your process to be scalable. You've had a number of different candidates in your drug substance selection phase. Maybe the best candidate in terms of PK data was not the best candidate in terms of manufacturability, but you've gone through that, you've selected that candidate. What do you need to know now? We need to talk about the formulation. Your CDMO is going to need to know what's in it. If you're going to have to dilute at your CDMO, we need to know what that formulation is consisting of. Make sure that they can supply the excipients that you need to perform that dilution step. At this stage, I would say try to avoid exotic excipients. Keep it as simple as possible. Less is more in this case. If your formulation can be stable with less exotic excipients, that will make your partner and future partners very grateful indeed. So your CDMO will have supply chain contracts with a lot of different suppliers, but let's make sure that they're able to supply the material you need and the quality you need to make sure that your drug substance can be filled successfully. In terms of success, they need to know a lot about the sensitivities of that product. They're going to want to define that design space for your process. They're going to develop a process, and if you don't let them know about any certain light sensitivity, temperature sensitivity, shear stress sensitivity that you know from a lab environment, that will maybe enable them to not put an optimal process together. So make sure you're communicating all those sensitivities to your CDMO partner early and in time to allow them to put together a process that will allow them to handle your product successfully without risking quality loss, but also making sure that you give them the information they need to put a process together that will enable the safety of their operators, to make sure that there's no risks on either end of the spectrum. Then after you've considered your drug substance and API aspects, the next step is selecting a container for that. So what is the right choice for your primary packaging combination? A lot of options are out there. And it seems they're getting more and more every day. Not just the glass suppliers are coming up with more and more innovative solutions for highly sensitive products. Also, stopper suppliers with different options around coating, non-coating, different rubber formulations, really tailored to meet what you need. Here, the same answer or the same question should be asked. What container is optimal for my product? Do I need specific coatings of stopper or vial? Is my product really so sensitive that I need to go into a niche or into an exotic component or container closure system in general? What components do you need? Are you going to be in a vial initially? I'd say in phase one, most of you will be landing in a vial initially, but also have one eye on the future. What is the right time to move into syringe? What is your end user preference? Are you looking more for a home care application? Does your modality allow for that? Or you're more in a healthcare environment, in which case you'll have a lot more flexibility. And how soon do you need to be in that injection device? If you are going into a home care system, you want it to be, usable for the patient at home. As I said, if you're going into that first clinical trial fill, you will most likely be in a vial. Make sure your partner has the ability to process what you need, the dosage you need, and the packaging combination that you need. But also think of one step further. It's never too early to start thinking about your end user at the end. So don't put roadblocks in place now that could damage or limit your options further down the road. As I said, patients are evolving. The needs of the patients are evolving. What they want to see, ease of use at home. These are all aspects that need to be considered whenever you're considering not just your phase one, but also your phase three material. But putting a nice package together with stability data collected early in the right container closure system can make out licensing way easier. So a lot of things to consider whenever you're putting together those decisions for container closure. As I said.
Another really critical thing to be thinking about early on is analytics around your drug product. Some of these are probably already set up your drug substance manufacturing facility. but how are you going to transfer these methods into your partner, and do you have robust methods in place that you can evaluate your approved attributes and your quality needs for your material? So
as I said,
kind of a high level example of some of the different analytical methods that we're typically seeing for early phase projects. And they're kind of grouped into a few different buckets. So there's really some incoming testing that typically needs to be done. And this is primarily around the drug substance, right? So the ID, the safety of it. We have some processes in place at Vetter at our clinical sites. This is clearly not universal, but we have processes in place where we don't necessarily have to do an ID test on incoming drug substance if it comes in a uniquely identified tamper and sealed container. But a lot of manufacturers, you might have to repeat that ID testing, confirming that the drug substance that they're going to be moving forward with has met all these qualifications to move forward. And then the next bucket is really that in-process testing. So here, what you're doing is you're testing any processing steps that your manufacturer is performing to make sure that the results and the outcomes are still accurate and are actually meeting the targets that you're searching for. So if your drug substance is a powdered API, for instance, or a frozen liquid that needs diluted, there's probably some type of processing step. So we need to transfer a method in, which is quite often a UV or a solo VPE, so that we can test and process to confirm that we're reaching these concentration levels that we're trying to. So we see that a lot. We also see a lot of density, pH, osmo in process testing. A lot of it up to the customer, but it depends a lot on the processing steps we're performing with material in our hands. And then as far as release testing, the only real requirement we as a clinical site have is for sterility. So we are ensuring that the product that we are passing off for the next step in the process to the clinic is in fact sterile coming off our lines. The rest of release testing is really up to customers, depending on what it is they're looking for, their release. We can transfer those methods in. Your partners in general can transfer those in, or you may already have them set up so samples can be pulled from manufacturing and sent to other third parties or your own facilities if you have these processes and methods already in place.
As I said,
some other key questions though, as you're talking about the methods. So here, I think once you have a plan in place, the question is, if you're looking to transfer these methods in, can your potential CDMO work with these methods? Do you have robust SOPs that can be transferred into their SOP systems, into their methods? Is equipment able to run and get the results you're looking for? So methods such as UVs and solo VPEs, they're also going to require some additional time to transfer in the methods. They're also going to require some representative material to make sure that the methods can effectively be run and the expected results can be obtained. So there's always time elements involved in these discussions. There's material involvement discussions to be had as well. So things to consider as you're planning analytics is definitely something that's going to require some thought and planning.
As I said,
so I alluded to it earlier. Gareth talked about it a little bit. I think one of the most important questions is the partner preparation and aligning the best partners and the partners that really complement your strengths and weaknesses with their strengths. So here's some of the questions, as you're starting to line up your different partners.
as I said.
So your partners should really be able to answer these questions, right? So can they safely handle your API? Do they have experience with your substance class? If you're bringing an oligo to the clinic, does your partner have experience handling powder APIs? Does their quality system, does it align with the expectations of your quality team? Their supply chain, their logistics teams, can they source the packaging you want? Is there a vial, as Gareth alluded to? Is there a syringe that you need? Do they have it? Do they have robust supply chains? Do they have backups? Can they perform your analytical methods? Do they have the instrumentation you need? So things we've spoken about a little bit already. And then ultimately, do the timelines match? Do they have capacity along the timelines that you need to meet? So timelines are always a concern. Lead times and sourcing materials are always critical components.
As I said,
another thing is, again, talking about timelines and partners. There are always contractual issues. There's quality agreements. There are audits that need to be performed. There are contractual obligations that need to be met. These all take significantly more time than you tend to think they will. So planning early for these also needs to be worked into the timelines. So once this is all done, once the quality agreements are signed, the contracts are signed, purchase orders are issued, what can you actually expect from this experience? So here, kind of a general, again, high level picture of what you can expect from a fill finish of an early phase 1, phase 2 clinical drug product for a liquid vial. So again, this is all kind of scope dependent. Syringes, lyos, there's a lot of different things that can impact these timelines. But generally, and again, very biased towards our experience at Vetter, from kickoff to your first batches are typically 3 to 6 months, again, depending on the scope of work going into that, if there are a lot of methods being transferred, if there are or are not technical batches, these can have impacts on timelines. But what's happening in those first couple months, really, again, it's these lab qualifications, it's getting materials set up in the system, it's getting your API set up as a material prepared with incoming inspections so it can be released for processing. It's procuring any supplies that are needed that are maybe not in stock or not currently on location. And this is all going into, in parallel, the tech transfer, which at Vetter is our process development specifications. This is the PDS. If you have or will work with Vetter, you'll see a PDS thrown around a lot. It's really the primary tech transfer document. It's really a step-by-step. All the materials, all the processes, hold times, stir times, really everything that goes into manufacturing your product goes into this. And this is what's discussed amongst project teams week after week leading up to manufacturing. And eventually, this is approved by both sides. And this is the precursor to the master batch records, which then tell our operators, it's the primary recipe for what they're going to be doing. In parallel with this, API, drug substance, there more often than not are challenges with shipments and logistics. So we do like to have the API on site 4 weeks prior to manufacturing. This allows us, again, to do our incoming inspections, to get things released prior to any manufacturing activities, which might start as early as a week before manufacturing. So a little bit of lead time is necessary here for the drug substance. So drug substance timing becomes a big issue as well for manufacturing.
As I said,
this is all ultimately leading up to potentially a technical batch. Ideally, there's always a technical batch. There's always things that we can learn and increase our chances of success and efficiencies and quality in our GMP clinical batches. Sometimes really easy, straightforward processes. It's not necessarily critical for a tech batch. Always preferred. If we are running a tech batch, then we also need 6 to 12 weeks in between so we can really review, digest, and incorporate findings from our technical batches into our clinical batches. So we're creating new PDSs, new batch records, new documentation based off of the results we got from the technical batches. And then ultimately, the GMP release of the batch to the clinic, or the Vetter release to the customer for the clinic. Our site anyways, our clinical sites, we're targeting 30 business days. So that gives our teams time to review and perform quality reviews of the executed batch records, all finalized testing and review and approvals of documents for sterility and any additional release testing we're performing. So overall, again, very hard to put an exact number on, but from kickoff to batch release, again, depending on the scope of the work, 6 to 10 months is a pretty good timeline for the manufacturing and release of a clinical batch. So a couple of final thoughts and questions as you're beginning this whole process. How much API is needed for this? So these are discussions to have with your partners. A lot of it depends on your intention with the batch, right? So you're going to the clinic, how many units do you need? And then you have to consider, are you pulling stability samples? And a multi-year stability test could actually dig into your material by quite a few pieces. So you always have to consider the sampling, any destructive sampling that's going to reduce your output as well. Rejection rates, line losses, things like that will be discussions to have with your partners to make sure that you're planning appropriately for your drug substance needs. So, and ultimately, if you're working with the right partners and you're putting this all together well, you're greatly increasing your chances of success in your clinical trial manufacturing.
So thank you, Mark. We might pivot just a little bit from, what are the overall, the general requirements and general things to give some thought to when you're planning that clinical trial material. And I'll have a look at a little bit closer what Fetter can do to support that along your journey. Fetter has literally comprehensive capabilities along the pathway and along the life cycle of that product through commercial manufacturing, assembly, packaging of devices, and getting that distributed to the world. But our focus today is on our Fetter development service. So that first step and that first interaction with what can be quite an intimidating and large company like Fetter. But we do have a strong development focus. And looking back just a little bit, Fetter began in 1950 in Ravensburg in southern Germany. In a pharmacy, still a family-owned company as of today, for the last 75 years. And so been really supporting pharma as a CMO for years, starting in 1965 with our first drug product manufacturing. And 2003 was when we really started that focus on development, changing our focus to a CDMO rather than a CMO, with the development of, or the introduction of our Fetter development services. A service in Ravensburg to help with product characterization before moving into that late stage clinical trial material manufacture and that sort of commercialization. And it was really around 2011 when that first major step was taken by putting our site in Skokie in Illinois, where Mark is based, on the map, solely for filling clinical trial material. As we know, the focus or the demands on clinical trial material, it is speed, it is efficiency. It's not so much about robust processes, it's about shortening that time to the clinic. So in direct competition with other quality by design aspects for a commercial product where you need robust processes that you really need to challenge the robustness of during development. So having a standalone site in Skokie really helped us push forward that clinical focus that we have. And we were lucky enough after about 10 years of having that site in Skokie, we were able to add to that offering that Fetter has by opening our site in Rankwell in Austria, where I am based. So we had the privilege of taking the process landscape that Mark and his team had put in place to support our customers, transferring that over to Austria, putting a European spin on it, adapting it to the site-specific things that we had there. And since then, we've been running very successfully, supporting, dozens, hundreds of customers with their clinical trial material since then. And, the next stage of that development will be kind of an upgrade to our US facility, moving from a shared facility in Skokie, Illinois, over to our own standalone site in Des Plaines. So only about 30 minutes away from where we are currently, with our own dedicated facility, with a lot of room for expansion to really build on what has already happened in Skokie, been built upon in Rankwell, and now the next evolution of that is going to happen in Des Plaines, and we're all obviously really excited for that. Where we fit in your network. So, nobody can do anything alone. That's the same in a lot of different branches of life, and Fetter fits right in the middle of your development pathway. So you've gone through your preclinical development, you've done some API candidate screening, candidate selection. You've gone through formulation development. You've identified that one candidate that will get you to where you want to be. You've developed methods to test it. Then you come to us, and we start supporting you with that process development to get that material into a container closure system that is suitable for that first in human trial. We go through, we support you with inspection, testing, release of that material with QP certification of that batch. And then we can support with our partners for that labeling and the next step of that let's say normal clinical trial distribution and performance. So we also have a lot of friends out there we work closely with that can support with that again. So Feather have commonly been known historically as a biologics focused company, and that was true for many, many years. But with new modalities that are coming out and a lot of really great investment and innovation in small molecules, peptides, oligonucleotides, we're seeing that shift with us as well. Antibodies and proteins are moving down to just about half of what we do now. Historically, it's been at least 3/4. And that number of oligos, peptides, and small molecules are really taking over our development side specifically. And we're starting to see those first molecules transition to our commercial sites as the approval gets met. So up to now, we've handled along both sites around 420 compounds we've processed. Audits are a key part of our day-to-day work. It's more or less every week we have an audit on site on one of our clinical sites. And, there are clinical projects ongoing along both sites from preclinical right the way up to phase 3 supply for around 160 different projects across both facilities. So, yeah, I could say we're quite experienced in what we do. And Feather, again, can be quite intimidating, but we're not just there for big pharma. Most of the customers we work with on our smaller sites are the small biotechs. We do have our big pharma companies in there with their pipeline, but the vast majority of who we work with have under 200 employees. So we're talking the small biotechs, the venture capital startups who have a molecule who want to get that first in human, to get a good phase 2 readout, to then explore licensing options. So don't be afraid. We're well attuned to working with our smaller biotech friends. And that brings us to the end of our slide presentation for today. That was a lot of information, I know. Thomas mentioned the slides will be shared with you. We can share a recording of this as well if you want to read back on that. But yeah, the key takeaways are, there's a lot of things that you can do today to help you streamline and ease the transition from the lab into the clinic. But there are also a lot of mistakes that can be made. So giving some thought to the key aspects you mentioned today will certainly help to ease that pathway, and choosing the right partners, for example, Feather, can also help improve your chances of success. So yeah, thanks for your attention today, and I'll hand over to Thomas.
That leaves me and us to say thank you very much for participating in this webcast. We hope that you got some insights. And yeah, as mentioned, please feel free to reach out to us to discuss your project. And yeah, so on behalf of Feather and the team, thank you very much and have a good day.
Thanks, everyone.